Okay, now we've covered the basics of how atoms and molecules work. Now we're going to dive down and go into detail about what they refer to as the molecules of life. In other words, organic molecules, molecules based on carbon atoms. And we like to start the chapter with some fun facts. So, why macromolecules matter? Chocolate. Okay, one of the most popular fun things out there. One of the reasons why it is so enjoyable is that it melts in your mouth. And why does that happen? Well, it's high in saturated fats, and saturated fats are solid at room temperature, but melt once warmed, and so that melting of fat in your mouth is part of what's referred to as mouthfeel and why it tastes so good. What does your hair have in common with steak? Well, they're both made of proteins, and proteins have all sorts of different tasks in living systems. There are structural proteins, like the proteins of hair. There are proteins involved with movement, like those in the muscles in that steak. And we'll be getting into proteins in a lot more detail later. Now, as you remember from the first chapter, the structure of your DNA is pretty much the same as that of a mosquito or elephant. We all have the same genetic language. And there's actually structures of DNA that will be basically alike in both a mosquito or an elephant or even a human. The differences in species is going to come down to the way the nucleotides are arranged, and we'll be spending whole chapters on how nucleotides work. Now, lactose. Lactose is the main sugar found in milk. Lactose intolerance is the inability to properly digest lactose. Lactose intolerance can be addressed by either avoiding foods that contain lactose or consuming lactase pills along with food. So something you'll see in a pattern here. A sugar often ends with OSE, so lactose is milk sugar. An enzyme often ends with ASE. So the enzyme, the protein that breaks down milk sugar, that breaks down lactose, is called lactase. So with lactase, even the lactose intolerant can enjoy the wonder of ice cream. Now, a cell is mostly water, right? I talked about aqueous solutions previously. So a cell is basically an aqueous solution with all sorts of reactions going on inside. The rest of the cell is mainly carbon-based molecules. And carbon can form large, complicated, and diverse molecules necessary for life to take place. Organic compounds are carbon-based molecules. Now, as I mentioned in the last chapter, carbon is a versatile molecule. Carbon can share electrons with other atoms in up to 4 covalent bonds. That's a huge diversity of carbon skeletons, varying in size and branching pattern, possible, because you don't have to have things in just a straight line, as we'll see in the next illustration. The carbon atoms of organic compounds can also bond with other elements, most commonly hydrogen, oxygen, and nitrogen. Remember, I said that much of what we'll talk about in this course comes down to 4, maybe 5 different elements. So this shows you some of the variation in carbon skeletons. So again, they can vary in length. If you're starting from the upper left, you can see a carbon skeleton chain that's only 2 carbons in length. You look to the one beneath that, and you can see that carbon skeletons can be branched or unbranched. So it doesn't have to just be a straight line. Moving to the other side, on the upper right, you can see that carbon skeletons may have double bonds. Remember, that's when 2 pairs of electrons are being shared. And those double bonds can vary in their location. And in the bottom right, you'll see that carbon skeletons may even be arranged in rings.
The carbon backbones of organic molecules may vary in length and in location of single and double bonds. Molecules may be straight or branched and may even contain rings of atoms.
So one of the simplest organic compounds is methane, which is written as CH4. So you have a single carbon atom bonded to 4 hydrogen atoms. Now, larger organic compounds are the main molecules in gasoline and are also important fuels in your own body. Because all those bonds holding together all those atoms represents what? A lot of energy. So being able to, you can store a lot of energy in these big complex molecules. And in the case of metabolism in living systems like us, there's a whole business to how do you break those bonds gradually and carefully so that you can harvest that energy that's being released as you do so. That's going to be a whole chapter later on, too. Again, you can represent atoms and molecules in different ways. There's structural formula, ball and stick model, and space filling models, just as we saw in the previous chapter. Now, it's odd to some people that we think of fuel in terms of dietary fuels for us as being like fuel for a combustion vehicle, but that's pretty much what it is. And you see that all those long chains, you don't really see the black carbon atoms as well, but you can see all the little hydrogens that are attached to those carbons. You can see that these are pretty long molecules. Now, the unique properties of any organic compound depend on the carbon skeleton and the atoms attached to that skeleton. So in an organic compound, the groups of atoms directly involved with chemical reactions are called functional groups. And many biological molecules will have 2 or more functional groups. On a molecular scale, many of life's molecules are gigantic, and that's why you see the term macro for large molecules. 3 categories of macromolecules are carbohydrates, proteins, and nucleic acids. Macromolecules are polymers. Polymers are large molecules made by stringing together many smaller molecules called monomers. A dehydration reaction links 2 monomers together and removes a molecule of water. Now, I like to say dehydration synthesis because this dehydration reaction is used to synthesize, to build larger molecules. The breakdown of polymers occurs by hydrolysis, by adding water to them, by inserting water at a point of a bond. That's going to be the reverse of the dehydration reaction. Instead of pulling out a molecule of water and using that energy to bind together 2 monomers or monomer to a growing polymer, you're actually sticking water in and splitting them apart. So here's a little illustration of that. So again, on the left-hand side, you can see a short polymer. On the right-hand side, you can see a monomer. And so to stick that monomer onto the growing polymer, dehydration synthesis is going to remove the components of water from each of those 2 reactants, if you will. And now that energy that was holding those parts on is used to then form the longer polymer. Going to the right-hand side, you're seeing the reverse. By inserting water, you are breaking a polymer chain. So just in a slightly larger illustration. And on to the animation.
Most biological molecules are very large and are built by assembling small molecules, or monomers, into long chains. The resulting molecules are called macromolecules, or polymers. A process of linking monomers, called dehydration synthesis, involves the removal of 2 hydrogen atoms and 1 oxygen atom to form water. One way this might happen is diagrammed here, where several generic monomers are shown with OH groups that could be used for linking. The animation illustrates the joining together of monomers by dehydration synthesis, catalyzed by a polymerase enzyme, and the reverse process, in which added water results in hydrolysis, catalyzed by a hydrolase enzyme.
Now, 4 categories of large biological molecules are found in all living creatures. The 3 I mentioned earlier, carbohydrates, proteins, and nucleic acids, and also lipids. Lipids are assembled a little differently, and that's why they're separated out from those other 3 guys. Now, carbohydrates include sugars and polymers of sugar. In animals, carbohydrates we often think of as dietary energy, but they're also raw material for manufacturing other organic compounds as well. In plants, carbohydrates also serve as a building material for much of the plant body, as well as a source of dietary energy. Monosaccharides, right, mono for one, are the monomers of carbohydrates. And so a monosaccharide can't get broken down into a smaller sugar. Right? These are the smallest units of sugars. Common examples of monosaccharides are glucose, you might see in sports drinks, and fructose, which is found in fruit. Both of these simple sugars are also found in honey. Now, glucose and fructose are interesting in that they're isomers. Iso refers to same. Isomers have the same molecular formula, but have different structures. So looking here at these 2 monosaccharides, so again, looking at the structural, looking at the formula rather, you see that both are C6H12O6. But those atoms are arranged differently. So if you look towards the top of these, and they've highlighted it for your convenience, you can see, oh, it's got the same parts, but they're not put together quite the same way. Which also means that they're going to behave differently in certain reactions. Now, the animation coming next is about isomers. We really are only going to discuss structural isomers in this class.
3 types of isomers are structural isomers, which differ in the covalent partnerships between their atoms. Geometric isomers, which vary in arrangement of atoms around a double bond. Enantiomers, or stereoisomers, which are molecules that are mirror images of each other, like left and right hands.
If you go on to take chemistry, you'll be dealing with those other types, but for this course, we're just really going to talk about structural isomers. Now, monosaccharides, particularly glucose, are the main fuels for cellular work. And we're going to spend a whole chapter talking about how glucose is broken down to yield energy for cellular work. In water, many monosaccharides will form rings. So, again, for the checkpoint, so all carbohydrates consist of one or more monomers, also called simple sugars. And again, so again, here's that ring structure of glucose. I talked about ring structures earlier on. Now, if you were to write out every individual atom in a glucose molecule, you'd see the illustrations that are in the first 2 from the left on this diagram. You can see that you can look at it in a linear form. In water, these monomers may form rings, and so you see that ring structure. But sometimes when you're writing out, you're writing out reactions using glucose, you don't want to spend half the day just writing in all these little guys. And so you'll see that abbreviated ring structure on the far right, where each angle represents a carbon atom, unless it's a different atom, like the oxygen that you see in the ring on the right-hand side. So it's just a quicker way of writing down a ring structure if you're working out how different molecules are working. All right, so every angle is a C except for where in this case there's an O. Now, disaccharides are constructed from 2 monosaccharides that have been joined by a dehydration reaction or dehydration synthesis. Disaccharides include lactose in milk, maltose in beer and malted milkshakes, and sucrose in table sugar. Sucrose is also the main carbohydrate in plant sap. So if you've ever had maple syrup, the plant sap is boiled to remove most of the water so that you concentrate that sap into a solution with lots of sucrose in it. High fructose corn syrup, abbreviated to HFCS, is made by a commercial process that converts the natural glucose found in corn syrup to much sweeter fructose. And that's, a lot of people think that it's just something you would extract, but no, it actually goes through a series of reactions involving genetically modified bacteria. And what happens is, because that glucose gets switched to much sweeter fructose, you wind up with something that tastes sweeter, which means you don't have to use as much to get the same level of sweetness. But unfortunately, it has some other side elements to it which have been associated with increased obesity. And hopefully I'll get a chance to describe how that works a little later on.
Sometimes organisms link sugar molecules in pairs to form disaccharides. Here are several examples. Plants make sucrose by joining glucose and fructose. Sucrose circulates in plant sap, and we obtain it from sugar cane and sugar beets and use it as table sugar. Lactose is formed by joining galactose and glucose. Lactose is the disaccharide that gives milk its sweet taste. Maltose consists of two linked glucose molecules. Digestion of starch in a sprouting seed or in the intestine of an animal produces this disaccharide.
Okay, so again, dehydration synthesis forms these double sugars. So monosaccharide plus monosaccharide equals disaccharide. And again, we have the dehydration synthesis. Now, sugar, the food labels years back didn't have as much detail as they do now. One good thing about the current form of food labels is that you can actually see how much sugar was added during production of the food item. Now, ideally, sugars added during manufacturing should account for less than 10% of the total daily calories, so that otherwise the sugars would be natural sugars that were present in, say, the fruit or the grain or whatever it was. Now, polysaccharides are complex carbohydrates and are made of long chains of sugars, so longer polymers that are constructed of monosaccharides. So it's like the counting system is 1, 2, and lots. So it's monosaccharide, disaccharide, and polysaccharides. Starch is a great example of a polysaccharide. It's made of long strings of glucose monomers and is used by plant cells to store energy, right? So we think of storing energy in our bodies in the form of lipids. Well, plants will store energy in the form of starch.
Polysaccharides are polymers, long chains consisting of hundreds to thousands of linked monosaccharides.
So you see here starch, glycogen, and cellulose. And at first glance, they look kind of similar, but then you look at them in closer detail and you realize, like, oh, okay, there are some differences as to how they're bound together. Now, looking at these three polysaccharides as you see them in the natural world, up at the top you have starch granules and potato tuber cells. So the formal term for starch is amylose. Again, there's that OSE ending. So amylose is these long chains, and plants will store them in cells, just like we store lipids in our cells, to serve as a long-term storage of food energy. Now, the one beneath it is one you may be less familiar with, glycogen. So just as plants have amylose in their cells for accessible energy, we have glycogen in our muscle tissue for quickly accessible energy. So when you say something where you have to suddenly run to catch a bus or something, you need a quick burst of energy that is right there, ready in the muscle tissue, as opposed to needing it to be delivered from the bloodstream, in the form of sugar or released from fats and all that. So it's a quick source of immediate energy right there in the muscle tissue. The third form is not a nutritive or nutrition polysaccharide, it's a structural polysaccharide, and that's cellulose. So looking at cellulose microfibers in a plant cell wall, and you zoom into that, and you can see that you have these long chains of polysaccharides, but they're cross-linked. You can see that the strands are held together by hydrogen bonds. So that makes it relatively tough. It also makes it harder to break down. In fact, animals don't make enzymes that can break down cellulose. To do so, animals that eat things and where they digest cellulose, they require microorganisms to do it. In our case, we don't effectively break down cellulose. That's why we refer to it as dietary fiber or roughage, because it's not going to get broken down and it's going to pass through relatively intact through the digestive system. So again, our starch granules, the glycogen found in human muscle tissue, and cellulose. So it just gives you an idea to see, how these things are assembled. You're not going to need to reproduce that on a test or anything like that, but just to get an idea that similar monomers, but put together in different ways. All right, so again, glycogen is used by animal cells to store energy and is broken down to release glucose when you need energy. So again, it's a quickly accessible form of energy stored in places like muscle tissue. Cellulose is really the most abundant organic compound on Earth, because think of all the plants out there. All those plant cell walls are made primarily of cellulose. If you look at them in close detail, you see that they form cable-like fibrils in the tough walls that surround a plant cell, and again, can't be broken down by any enzyme produced by animals. So here you can, again, the major themes, I'm going to tend to skip over these in the lectures and allow you to sort of think about these as examples for yourself. All right, lipids. So first carbohydrates, second here are lipids. Now, almost all carbohydrates are hydrophilic. Hydro meaning water, philic meaning loving. So hydrophilic means something that dissolves easily in water. Lipids are hydrophobic, so water-fearing or water-hating sometimes is the other interpretation of phobic. So things that are hydrophobic don't mix with water. Now, lipids differ from carbohydrates, proteins, and nucleic acids in that neither are they huge macromolecules, nor are they necessarily polymers built from repeating monomers. So that's why we tend to separate them out from those other three types. But they are a diverse group of molecules made from different molecular building blocks. And again, most of us are familiar with the fact that you can't mix oil and vinegar if you're making something like a salad dressing, that if you shake it up and let it sit for a while, they will separate out. That's because vinegar is basically an aqueous solution, and oil will not mix with that, so you see that layering form. Now, most of us think lipids, we think fats. A typical fat or triglyceride consists of a glycerol molecule joined with 3 fatty acid molecules via dehydration reaction or dehydration synthesis. So triglyceride is glycerol plus 3. That's what the tri comes from, the 3 fatty acid molecules. And they perform important functions in bodies like our own, including energy storage, cushioning, and insulation. And you tend not to think of the cushioning so much, but if you look at the human kidney, most of your important parts are kept in bone cages, right? If you think of your skull, right, all the major sensory organs and of course the brain and all that, the spinal cord is contained within the backbone. Your heart and lungs are contained within a rib cage. The lower reproductive organs and such are sort of contained within the pelvic girdle. In between the rib cage and the pelvic girdle, along your dorsal surface, on your back surface, is where your kidneys are found. Your kidneys are beneath the surface of the skin, of course, but they're not contained in a nice, safe, bony cage. So what is to prevent these really critical organs from mechanical damage, say, bumping into things, is that they have a layer of fat that surrounds them. And that serves to cushion them from possible damage from external forces. Insulation, if you've ever seen something like a whale or a seal, one of the reasons why they have all that fat is to insulate their bodies, to not lose heat to the cold waters in which they travel. So here we see the synthesis and structure of a triglyceride. So again, there's your glycerol, that vertical rectangle in the diagram. And on the top diagram, you see a fatty acid joined to it by dehydration synthesis. Well, repeat that 3 times and you wind up with a fat molecule. So it's a glycerol head and 3 energy-rich hydrocarbon fatty acid tails. And you notice that one of them has a little kink in it. We'll get to that in a little bit. So again, dehydration synthesis, repeated 3 times, and now you have a triglyceride.
Butter, lard, margarine, and salad oil are composed of lipids called fats. To make a fat molecule, 3 fatty acids bind to a molecule of glycerol. For this reason, fat molecules are technically called triglycerides. Fat molecules do not mix with water because they have 3 long, nonpolar hydrocarbon tails. Cells use fats for energy storage because the tails hold more potential energy than other biological molecules. The fatty acid tails in a fat can vary in length. A very important characteristic of fats is the number of double bonds in the hydrocarbon tails. Fats that contain only single bonds are called saturated fats. Fats containing double bonds in one or more tails are called unsaturated fats. Unsaturated fats tend to be liquid at room temperature. Vegetable oils are examples. Saturated fats, like butter and lard, are solid at room temperature. Saturated fats in the diet can lead to heart disease, while unsaturated fats are safer.
So again, you see that the double bond causes a kink in those lines. So I'd say, well, what does saturated mean? You think of saturated as in terms of water, right? You go out in the rain and you get soaked and you can't get one more drop of water, into your clothing or in your hair or whatever. That means you're saturated, right? You are filled with water. You're covered with water, and any additional water is just going to drip off of you. Well, think of saturation in this case as hydrogens. If you can't fit one more hydrogen on that carbon chain, then it's saturated, right? It's saturated with hydrogens. If you see a double bond, that means that potentially there could still be hydrogens added to it. And that's why they say unsaturated. So if that carbon skeleton has fewer than the maximum number of hydrogens at that double bond, it's unsaturated. If it has the maximum number of hydrogens, it's saturated. So saturated fat has all 3 of its fatty acids saturated. You might say, well, what does that mean for anything? Well, go back to this illustration here. Oh, sorry. That's going to
butter, lard.
Sorry, apologies. If you look at these 3 tails, all right, the 3 tails in this illustration are saturated. You notice that they're nice and straight. Well, the fact that they're nice and straight means what? You can stack them up easily. And so you wind up with that relatively stable form and that business of being solid at room temperature, whereas if there's unsaturated fatty acid tails, you get that kinking, and that means that it's not going to stack up nice and neatly. It's not going to be solid at room temperature. It's also going to be a little less stable. So here we talk about relative amounts of different fats in different food items. So corn oil is mostly polysaturated. What they mean by that is there's more than one double bond, right? So if you have a single double bond in forming that one little kink, we call that monosaturated. If there are many double bonds, then that means it's polyunsaturated. So something like corn oil, which is made of both primarily of poly and monounsaturated fats, it's going to be liquid at room temperature. Palm oil is kind of interesting. It's a plant fat, but it's 50% saturated. So you can get the stability, it doesn't go bad as quickly. You have the stability and the room temperature solid elements that we tend to think of as animal fats, but it's in a plant fat. And one of the reasons why you see it so often is that it sort of works in a sense like butter or lard would, but it's of plant origin. So that's why you'll see it in a lot of things like cookies and pastries and things like that. Butter, on the other hand, is an animal-sourced fat, right? It's made from milk. And you can see it's 58% saturated, right? So even more double bonds in the fatty acid tails. And only a small amount of it is either mono or unsaturated. So most animal fats have a relatively high proportion of saturated fatty acids. Again, they can easily stack because you don't have those kinks, right? So they're nice and straight. You can stack those up, and that means it will tend to be solid at room temperature. Unfortunately, that also means that they can contribute to arteriosclerosis. Okay, so when we're talking about this, scler, S-C-L-E-R, means hard or stone-like. And so these fats that can build up plaques on the blood vessels makes them harder. And if blood vessels can't stretch, it means that it's going to lead to blood pressure being higher, because stretching of blood vessels takes up some of that force, some of that pressure from the beat of the heart. If you can't stretch, that means that you can't release some of that energy in stretching, and you wind up with things like high blood pressure. Now, most plant and fish fats tend to be high in unsaturated fatty acids and therefore liquid at room temperature. You might say, well, wait a second, I've seen things like hydrogenated corn oil. Okay, hydrogenation is adding hydrogen. So you can convert an unsaturated fat to a saturated fat. So, in other words, make a liquid fat solid at room temperature and somewhat more stable over time. But in doing this, you create something called trans fats, which is a type of unsaturated fat that is really bad for your health. So you're really better off eating naturally saturated fats, typically animal sourced fats, than saying, oh, here's a plant oil, this is healthier. But if it's been hydrogenated, then that means it's not healthier. So yes, it does turn a liquid fat into a solid fat, but it creates a type of saturated fat that's really not good for human bodies. Right. So again, saturated fats typically animal origin, unsaturated fats typically plant origin, or from fish sources. And again, looking at the text underneath the donut there, you can see soybean oil, fully hydrogenated cottonseed oil, partially hydrogenated cottonseed oil. So fully hydrogenated means it is a completely saturated fat. Partially hydrogenated means that it's slightly it's not completely hydrogenated, it's partially hydrogenated, which means that there's going to be some of those bonds would still be kinking, right? You'd have some of those double bonds remaining. Now, steroids are lipids, but they're very different from fats in structure and function. For one thing, a steroid has a carbon skeleton with 4 fused rings. What varies one steroid from another are the functional groups attached to that set of rings, and those chemical variations will affect how they function. Now, cholesterol is a key component of your cell membranes and is the base steroid from which your body will produce other steroids like estrogen and testosterone. Also, we tend to be taught that cholesterol is bad. Not all cholesterol is quote-unquote bad. In fact, your body will construct cholesterol if it has to. So don't keep in mind that there are some cholesterols that are relatively healthy and some which are less healthy. Now, here's a couple of examples of steroids. Again, looking at the top, you have that base steroid cholesterol. You see the 4 fused rings and some functional groups hanging off of that. Well, the body can convert that base steroid to either testosterone. You can see that you've sort of edited off a lot of the upper tail there, or a type of estrogen, or you've also done some editing to that base cholesterol. So from cholesterol, the body can edit the functional groups attached to it to make different steroids that will have very different functions. All right, so one of those would be things like sexual dimorphism. And one thing that you may have heard the term steroid before, and steroids are sometimes discussed in terms of sports, right? Synthetic anabolic steroids. Now, a couple of terms here. Anabolic. When you talk about an anabolic reaction, right? In metabolism, you have building reactions and breaking reactions, right? So reactions are you building things up, reactions are you breaking things down. Anabolic reactions are those where you're building something up. So synthetic anabolic steroids are variants of testosterone. They mimic some of the effects because they have similar functional groups on them. And there are medical uses for these, treating diseases like cancer and AIDS, but they can be abused by athletes to build up their muscles quickly, right? Anabolic, building. So instead of working out and eating right and all the rest of it, athletes could, some athletes try to cheat by taking these drugs that makes the body build up their muscles quickly, right? Without the usual stress, using the usual stress of exercise to do so. Problem is when you have things like hormones, and testosterone is an example of a hormone, hormones often have more than one precise function. There's often other things going on. And the downside of doing things like building up your muscles quickly by using synthetic anabolic steroids is that you also have things like aggression that increase because testosterone is associated with aggression. So you wind up with some serious, challenges, like sometimes you might have heard the expression roid rage, right? People using steroids having, relatively labile, rapidly changing emotions. There's also physical problems. The body is full of feedback systems, right? Things that measure stuff like temperature, blood sugar, etc. Well, the body also measures how much testosterone is in it. So if you're a male and you're normally your body has got a feedback saying, okay, we've got so much testosterone, we got to secrete some, okay, don't secrete so much. If you start piling these testosterone-like chemicals in the body, the brain sends out the message like, oh, we've got enough, we don't need to make more. And the body will, as you add testosterone to the person, their body is progressively making less and less testosterone of its own. And the result is what I heard referred to by a wrestler as it turns the grapes to raisins. And that you get atrophication of the testes because the body is trying to turn down the testosterone production, not knowing that's being added from the outside world. So most athletic organisms ban these. And every edition of the book, we unfortunately have some new athlete being presented. And so in this case, I have forgotten the man's name, but he was renowned as a great batter. Well, he was also renowned for abusing steroids. Okay, so carbohydrates, lipids, now proteins. Proteins are polymers of amino acid monomers, right? So now we're back to a polymer system. Proteins account for more than 50% of the dry weight of most cells, and they're important in pretty much every function of cells. So you see here a few examples. What I'm going to do is go through each in turn in the subsequent illustrations. So structural proteins, they provide support. So the hair, I mentioned in the first part of the semester, first part of the lecture, but also things like ligaments, right? The things that attach muscles to bone, horns. As you see the horns on the sheep, they're primarily proteins. In the case of horns, it's proteins coating a bony core. In the case of antlers, you'd see antlers get shed by deer. Well, that's because they're not on a bony core, right? So you've got this basically tightly compressed hair-like material, and then the animal can just shed it off and grow a new one the following year. Storage proteins, right? We think of eating things for proteins. One of the reasons we eat things like eggs and seeds is to get protein. Why? Well, in both these cases, these are mechanisms for storing proteins for the use of a developing embryo, be that of a plant or a reptile here. So either way, the amino acids are going to be broken down for growth of a growing individual organism. So that's why we consume them, because we can take these storage proteins and digest them to provide amino acids for our own uses. Contractile proteins, right? So that, referring back to that steak in the beginning of the chapter. So contractile proteins are associated with movement. So this will enable muscles to contract. Muscles operate by getting shorter. Muscles don't push to get longer. They get stretched longer by some opposing force. So contractile proteins, their job is to contract. If you take A&P, you will learn the mechanics of this in great detail. Transport proteins. There are proteins whose job it is to move stuff around. And the protein hemoglobin found in red blood cells is there to transport oxygen. And one that we'll spend even more time on are enzymes. Every time you see a chemical reaction, like the dehydration synthesis earlier and the hydrolysis reactions, you saw that blob in the illustration enabling those reactions to take place. Those were enzymes. Enzymes help chemical reactions take place without becoming part of the product. The way I like to think of this is, let's say you go to a party and there's people circulating, well, pre-COVID, okay? You go to a party and people would be sort of milling around it, and you might meet someone on the other side of the room, but it might take all evening for you to start wandering around for you both to run into each other. Now, often there's that one person at a party who will say, oh, you've got to meet this other person. And they grab you by the arm and they drag you over to meet this other person and introduce you. And maybe you hit it off, maybe you get to, chatting, whatever. But say things, blossom into a friendship or a relationship, that person who introduced you is not going to form, a bond with those two people. They're going to go off because they're going to introduce two other people at the party. So you might have eventually met each other, but that person made the meeting happen faster. And that's essentially what enzymes do. They catalyze, and that's what they mean by catalysis, or something that catalyzes. Things that make a reaction happen faster because it takes less energy to do so. We'll get into the details of that a little later on. But the enzyme helps the reaction without becoming part of the product. So the enzyme will get the reactants together, but then it'll leave to join other reactants in other chemical reactions. Okay, so proteins. This is another polymer. So all proteins are made by stringing together a common set of 20 kinds of amino acids. So the amino acids are the monomers that are joined together to form the polymers we call proteins. Now, each amino acid, each of these monomers, consists of a central carbon atom bonded to 4 covalent partners. 3 of those attachment groups are common to all amino acids, right? A carboxyl group, an amino group, and a hydrogen atom. So you've got 20 kinds of amino acids. All 20 have those 3 attachment groups. The ones that is different is called a side chain. So each type of amino acid has a unique side chain, which gives that amino acid its special chemical properties. Right, so I think I have an animation coming up in a couple of slides, but you see here, let me go to the larger view. So there's your central carbon, there's the hydrogen, there's the amino group, there's the carboxyl group, and the part that varies is represented by the little orange square there. So all amino acids are going to have that first top part the same. The part that varies is going to be the side chain. So for example, we have leucine and serine here. So again, that whole top part is the same for both amino acids. The part that's different is the side chain. And one reason why the side chains are important is that they can have different characteristics. For example, here we have an amino acid that has a hydrophobic side chain, and then we have another which has a hydrophilic side chain. So they're going to behave differently in reactions because they have those different properties. So that side chain is important as to how the amino acid functions. So cells link to amino acids together by dehydration synthesis, just as we saw with the carbohydrates. We call the bonds that join amino acids together peptide bonds. So a long chain of amino acids is also called a polypeptide. Poly, many, peptide referring to the peptide bonds. A functional protein is one or more polypeptide chains that is twisted, folded, and coiled in a very specific way into a molecule of a unique shape. So here again, you see the two amino acids. Watch what's happening at the two ends that are meeting together. And again, dehydration synthesis. You're removing the OH group and the hydrogen, and now you're binding together those two carbons at the point where they were. So that bond from carbon to nitrogen is referred to as a peptide bond.
Most proteins are folded into a complex globular shape. Each protein molecule consists of one or more chains of amino acid monomers. The amino acids are linked by peptide bonds, so a protein polymer is often called a polypeptide. Because they are so complicated, proteins are usually described in terms of 4 levels of structure.
And the first one you see here is the primary level of structure, and that's just the sequence of amino acids, right? So first, second, third, fourth, etc. Because that will be the same in a particular protein. So you have one enzyme of lactase, one molecule of lactase, another molecule of lactase. They're going to have the same sequence of amino acids. Now, proteins use those 20 amino acids, think of them as letters, to create polypeptides, words that are hundreds or even thousands of amino acids in length. So the amino acid sequence of each polypeptide is going to determine the three-dimensional structure. The three-dimensional structure of the protein enables the molecule to carry out its very specific function. Nearly all proteins work by recognizing and binding to some other molecule. So the shape of that protein and the presence of things like hydrophobic, hydrophilic, side groups are going to determine how something is going to interact with, that protein will interact with another molecule. So again, there's that primary sequence, right? They show the amino acid sequence, in this case of lysozyme. But that's just the primary sequence. That's just the chain of amino acids. What's going to happen next is that chain is either going to be folded or coiled. You can see the coils in the upper right-hand side. And the coils are kind of like a telephone cord, if you can remember back in the old days when we had landlines. And then the folds are kind of harder to distinguish, but it's like you take a piece of paper and you fold it so that it forms like an accordion shape, or a pleated shape. So that would be the secondary level of organization. Now, that is going to then undergo folding. That's your tertiary or third level of organization. So let me pop through these in more sequence. So again, primary organization, secondary here on the left. So these are either the folds or coils. And that model sort of shows you in detail, right, with all the atoms involved on the right-hand side.
Each protein has a unique primary structure, a particular number and sequence of amino acids making up the polypeptide chain. 20 different amino acids are used to build proteins. Theoretically, the various amino acids could be linked in almost any sequence, forming an almost infinite variety of different proteins. This illustration shows some of the amino acids making up the primary structure of a protein. The structure of a single generalized amino acid is shown below. The main backbone of every amino acid is the same. This is what forms the backbone of the polypeptide chain. It is the R group, which projects out from the backbone, that makes each of the 20 kinds of amino acids unique. Different amino acids have different properties that affect the folding of a protein. Thus, primary structure ultimately determines the shape of a protein, which determines its function.
Okay, so a slight change in the amino acid sequence can affect its ability to function, right? Because, again, thinking of things like those hydrophobic, hydrophilic side chains. If you have a different arrangement of amino acids, it means that you might have an area that's hydrophilic when it should be hydrophobic, or vice versa. So it can't fold, coil, or it can't be folded, coiled, or compiled, folded up into the same shape as a normal one. And one well-known example is the substitution of one amino acid for another at a particular position in hemoglobin. That's the blood protein that carries oxygen. And that one change of an amino acid for another means that the protein has an incorrect shape. In fact, it crystallizes and forms something like a curve. That's where the sickle part of sickle cell disease comes from. And it's an inherited blood disorder. There's a mistake in the gene, a mistake in the recipe, if you will, for making that protein. So if we look at the illustration here, you see that those two chains look the same, but you have the substitution at one point in the chain. I'll show it in another slide. Well, the normal polypeptide has that certain blobby shape, which is found in a normal red blood cell. But you can see, using the molecule model, that sickle-shaped polypeptide is not shaped the same way as the normal polypeptide. And you wind up with that sickled red blood cell. So again, normal polypeptide, which can normally function normally in a red blood cell, versus the sickle-celled one. And if you look closely at the top of the amino acid sequence, you'll see that the 1, 2, 3, 4, 5, 6th amino acid is shaded red. Now compare that to the one in the previous one. It says valine in there, or VAL. 1, 2, 3, 4, 5, 6. Oh, that says glue for glutamine. So you've substituted that one amino acid, and it causes that radical change in form. So one difference in structure is enough to cause the protein to fold into a different shape. That alters its function, which in turn can result in disease. And misfolded proteins are associated with many diseases, including some of the serious nervous system disorders. The diseases shown in the next slide are all caused by prions. Prions are misfolded versions of normal brain proteins. Prions are really bizarre, because we think of living things as having certain characteristics. Well, a prion is simply a misfolded protein, but somehow prions can make normal brain proteins fold incorrectly. So a prion is a misfolded protein, but it's also contagious. It causes other brain proteins to become misfolded. So you see here, this is what's shown here is something like. I'm going to see you tomorrow morning starting at 8 a.m. Eastern. So prions are involved with something like mad cow disease, or what you see in that poor moose there, which is a wasting disease. And again, this is fairly bizarre. You have, looking in the brain illustration starting on the left, you have normal proteins, and then you have the prions, which are the V-shaped guys, which are misfolded proteins. And somehow they convert the normal proteins into these misfolded prion proteins. Well, those proteins clump together. And so certain disorders are associated with prions, and that's, we talk about mad cow disease. That's an example of that. And it takes a while for that damage to progress. So one of the reasons why people were concerned about eating meat that might have been contaminated by nervous tissue containing prions was that you could have these prions get into, say, a human brain and begin converting the proteins, and over time lead to damaging many brain proteins and leading to things like, disorders like what we commonly call mad cow disease. And again, it's something that we don't really have a treatment for. We don't really have a means of undoing this damage. So it's something that can be detrimental, and in many cases, in things like these wasting diseases and ungulates, it can be lethal. Now, protein shape is sensitive to the environment. So an unfavorable change in temperature, pH, or some other factor can cause a protein to unravel. So when you see that complex folding of a protein, one of the things that holds those folds together are hydrogen bonds. So if you have an unfavorable change in temperature, that can break the bonds. pH, what is pH but a measure of whether something is taking up hydrogens or giving off hydrogens, right? So taking up hydrogens are bases, giving off hydrogens are acids. Well, those can lead to reactions that cause the protein to unravel. And we're actually going to have a lab where we get into this in detail. Okay, last group, nucleic acids. Nucleic acids are another monomer polymer type of organic molecule. These are macromolecules that store information. And that's it. They're a one-trick pony. It's an important pony, but they don't do things like form structures, store energy for food, anything like that. Their job is just storing information. And they're storing information that provides the instructions for building proteins. So when you saw those amino acids in those proteins in the last section, well, what determines that sequence of amino acids? The instructions that are contained in nucleic acids. Now, there's two types of nucleic acids, DNA and RNA, and they're both abbreviations, right? So DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. And what those refer to is essential sugar in the structure of those nucleotides. So the genetic material that humans and all other organisms inherited from their parents consists of huge molecules of DNA. And the DNA resides in the cell as one or more very long fibers called chromosomes. Chromo meaning color, some meaning body. So literally colored bodies. These were some of the first things that people were able to see under the microscope, and they stain very easily. So people were starting to look at things like plant cells or animal cells or whatever, and they were staining them so they could see them in more detail. And what took up the stain was visible most easily in some of these early preparations were the chromosomes, so hence the name colored bodies. Anyway, a gene is a unit of inheritance encoded in a specific stretch of DNA that programs the amino acid sequence of a polypeptide. So you could think of genes as essentially recipes, right? It's an individual unit of information to create a specific amino acid sequence. And we'll get into, in far more detail later on, the whole business of how does information go from a gene, a specific stretch of DNA, to a protein. Well, you have a go-between called RNA. So DNA has the information. One type of RNA we'll talk about, its job is to take a copy of that information, and that information is then used to assemble proteins. Now, nucleic acids are polymers made from monomers called nucleotides. So you had monomers, monosaccharides and polysaccharides. You had amino acids and polypeptides. Now we have nucleotides and nucleic acids. Each nucleotide has 3 parts. So just like we had the amino acids where 3 of the 4 parts were the same and the only part that differed was the side group, here we have a 5-carbon sugar and a phosphate group that will be the same. What's going to vary will be the nitrogen-containing base. So seen here, you see that this 3-part molecule, the phosphate group illustrated in yellow and the sugar illustrated in blue, that part's going to be the same in any DNA nucleotide. And that sugar is the deoxyribose that is part of the name. The part that's going to be different is that nitrogenous base represented in green in this illustration. So the symbol used in the book is showing the sugar phosphate, what's going to be part of the backbone of the DNA molecule, and then you see that base sort of hanging off in the middle. For some reason, the sugar illustration, the sugar should be pointing to the blue box, not the green forked thing. Okay, so again, the phosphate and the sugar are going to be what connect one nucleotide to the next. The nitrogenous base is sort of hanging off in the middle of this. Now, here we go, so there's a better representation. So phosphate and sugar, those would be the parts that attach to each other, forming the chain of the chain of nucleotides forming the nucleic acid, and the bases will come off in the middle. We're going to be talking about these guys in great detail in the next couple of chapters after this. So each DNA nucleotide is one of 4 possible nitrogenous bases, so this is like the 20 possible side groups that you saw in amino acids. And here they are. So the nitrogenous bases, you'll see that 2 are double rings and 2 are single rings. Now, because when they bind together and wind around each other, the 2 strands that form your typical DNA molecule, you'll notice that it's sort of the same diameter all the way down. So in pairing, you have to be pairing one double ring with a single ring, right? Because if you had pairs of double rings and pairs of single rings, then the thing would be, wide and narrow and wide and narrow. So the pairing is going to be single to double, single to double. In this case, it's going to be adenine to thymine, guanine to cytosine. And the way I remembered it back when dinosaurs walked the earth was that adenine and thymine both had letters made by straight lines, and guanine and cytosine have letters that are made with curved lines. Now, it's going to break down when we get to RNA, but it works at least for DNA. So there's your 4 nitrogenous bases. And how are they assembled? Dehydration synthesis, right? So dehydration reactions link nucleotide monomers into long chains called polynucleotides. These are forming covalent bonds between the sugar of one nucleotide and the phosphate of the next. So the fact that they all join together, they collectively form a sugar-phosphate backbone. And the bases hang off that backbone like appendages. We'll see this in better detail in a moment. So a molecule of cellular DNA is double-stranded. You have 2 polynucleotides strands coiled around each other to form a double helix. And this is a pretty classic illustration now. When people talk about science, you tend to see microscopes and DNA helices. So bases along one DNA strand are going to hydrogen bond to bases along the other strand. So it's almost like a ladder, right? Where the sides of the ladder is the sugar phosphate backbone. The rungs are going to be the 2 bases binding to each other in the middle. So again, they pair up specifically. A only pairs with T, G only pairs with C. So if one DNA strand has the sequence GAA, TGC, what would be the sequence of the other strand? It would be CTT, ACG. So again, here's that single strand of DNA, and then on the left-hand side, the double strand, and now you can see that the bases are binding with hydrogen bonds in the middle. So again, it's kind of like a ladder that's been twisted. So repeating sugar phosphate backbone, nucleotides, bases hanging off that. Each unit of sugar phosphate backbone plus a base is a nucleotide, so this is a chain of nucleotides. In other words, a polynucleotide. Well, a DNA molecule is two polynucleotide strands wrapped around each other. Now, both DNA and RNA are polymers of nucleotides, and both are made of nucleotides containing a sugar or phosphate in the base. But there are differences between DNA and RNA. As you can tell from the first letter, the sugar is different, right? RNA has ribose instead of the deoxyribose that's in DNA. So that's one difference between DNA and RNA. Second is, instead of the base thymine, RNA has a similar but different base called uracil. That's why the straight letter, curved letter thing breaks down when you get to RNA. Also, RNA is usually found in a single-stranded form, whereas DNA usually exists as a double helix. So now looking at an RNA nucleotide, again, we have a sugar phosphate backbone, but now the sugar is different. And we have the nitrogenous base, which in the case of A, G, and C are the same, but instead of the T of DNA, you have the U in RNA. Okay, so hopping over the seams. There. Okay, so they like to give examples of actual science in these chapters. This is a good question. Does lactose intolerance have a genetic basis? So, giving you a little background, most lactose intolerant people have a normal version of the lactase gene. So why is it that these people, have lactose intolerance? Why is there, what is the genetic basis for this if the gene itself is normal? Well, people want to study, populations to see what the chromosomes are like in lactose intolerant versus lactose tolerant people. Now, they know that lactose intolerance can be correlated with a single nucleotide at a particular spot within one chromosome. And to test this hypothesis about this one nucleotide, researchers examined the chromosomes of 196 lactose-intolerant people from 9 families, right? So it's not just one family, there's multiple families to make sure that it isn't something specific to that one family. The cool thing is that the results showed 100% correlation between lactose intolerance and having cytosine versus thymine at this one particular spot on a chromosome. So this is interesting. So it's showing you how a small change in a DNA nucleotide sequence outside of a gene can affect production of a protein, and in this case, the well-being of the organism. So we always think that it's got to be a change in a gene changing a protein. There's changes in DNA that are not changing the sequence of a protein, but they do have influence on what's going on with that gene. So giving an example here, so they show you the lactase enzyme. That's actually one of the authors that's in that little cartoon on the bottom there. And I assume he's lactose intolerant from this illustration. Looking at the DNA, on chromosome 2, you see that black bar representing the lactase gene, and 14,000 nucleotides further down the chain is the site of that one nucleotide associated with lactose intolerance. So in a person who cannot digest lactose, it's cytosine. In a person who can tolerate lactose, it's thymine. So again, here's that illustration of the enzyme itself. Here's the individual, their DNA. And again, on chromosome 2 in the human, human genome, there's that one spot where you find the lactose gene, and then way further down the line is the site with that specific nucleotide where substituting cytosine for thymine means that the individual will not be able to digest lactose. Now, most people are lactose intolerant as adults, because if you think about it, lactose, is in milk. Normally, we would feed on milk only as a very small person, right? As a baby, the person's getting milk as a nutrient, source from their mother. But you stop doing that, so it would make sense that lactose tolerance would not be that important later in life. Unless, of course, you have a culture where you are reliant on dairy products. Now, lactose intolerance is found in 80% of African Americans and Native Americans, 90% of Asian Americans, but only 10% of Americans of Northern European descent. Well, how is this? Well, natural selection. In cultures where dairy products were a staple in the diet, natural selection would have favored anyone with a mutation that kept the lactase gene switched on past infanthood. And we'll get into how genes are switched on and off later on. But so the lactase gene is normal. What's abnormal in the case of people who can digest lactose past infancy is that mutation, right? So in a place where getting nutrients from dairy products was important, if you think of some of the northern climes where crops are not going to be around in the winter, or in certain desert areas where a primary source of food will be these animals that travel with you. Well, that natural selection would have favored anyone who could still digest milk. So there's ethnic groups in East Africa that rely on dairy products. They're also lactose tolerant, but it's due to 3 other genetic changes that keep the lactase gene permanently active. So it's fascinating in that you have the same result, tolerance for digesting lactose, but in Northern Europeans and East Africans, it's actually 2 different mechanisms. It's 3 genetic changes in the folks in East Africa. It's only 1 genetic change for the people from Northern Europe. So it's kind of fascinating. You wound up with the same result by 2 different genetic change, 2 different sets of genetic change. So for those folks who rely on mammals to provide milk, there's more than one way to digest a lactose molecule. And that's the end of the chapter.